Hi, everyone.
Hello.
Hey, Dr. Allen.
Hello.
Nice to see everybody in the virtual world here. So just getting a few more students logged in and we'll get started.
Dr. Todd, can you hear me?
Hi, Elena, I see you, yes.
Hi, I'm traveling, so I'm gonna be off camera, but I will be in, I just got caught up. I think I'll be home, sooner. So I'm just gonna join in while I'm on the ride.
Okay, just as long as you're not trying to drive at the same time.
No, I'm not driving. I jumped in the passenger side.
Okay, all right, thanks for being here. Okay, let me take a look at some chat notes. Yes, okay, great. All right, folks, welcome back. And let's get started here. So I should be starting chapter 4 today, but I'm not quite ready to do that. So our goal here is to finish chapter 2 today. If I have the luxury of time at the end of today's lecture, I might start chapter 4. And I might even start a little bit of lecture of chapter 4 in lab for like 20-30 minutes. So we'll see how the afternoon goes. But welcome back. So good to see you. I hope you're doing well. I know there may have been some questions about, we did have some Dropbox challenges and things like that. We can talk about any of those issues as we get into lab today, but I'm pretty confident that I do have just about everybody's submitted work. So I think it is beginning to work as it's supposed to, okay? So we'll talk about those things in lab in just a little bit. Okay, do you have any questions?
Just briefly on the lab Dropbox. Is that the Dropbox we'll be using for all future lab assignments as well?
It will be, yes, Taylor, yes.
All right, thank you.
Okay. So yeah, I mean, once we verify that that Dropbox link is working, maybe you want to bookmark that in a web browser or something like that so you have easy access to it. Okay, let's begin here. I want to share my screen with you. And bring up what we were talking about last time. Okay, so these are new notes, but what I've done is copy the last portion of last lecture's notes. We were talking about covalent bonds. Okay, so we were in the topic of chemical bonds in general. If you have your textbooks available, I'm talking about the information on pages 27, 28, 29, this type of thing. Okay, so we're going to continue right here. We were talking about covalent bonds. These are bonds where the attraction is created between atoms that are sharing electrons. And we mentioned that there's two varieties, polar and non-polar covalent bonds. Okay. In your textbook, the only picture, image, or graphic of covalent bonds is figure 2.3 on page 29. It's useful, but I think I can share some other things with you today. We can do a little bit better than that. Okay, so let me just clarify what we're seeing here on the drawing. Okay, so on the drawing, the dotted dash line here is trying, oh, that was ugly. Let's do that again. Sorry, folks. Technology is magnificent. Okay. All right. Okay, so the dotted dash line is supposed to represent the motion of shared electrons. Okay, just so we get our minds warmed up again to what the conversation really was. And so you notice that with a nonpolar bond, you get roughly an equal sharing of the electrons. And what I mean by that, an electron is not like a pizza. You can't, with a pizza, you can cut a pizza in half and give half of it to your sister and half of it stays with yourself, right? We're not cutting electrons here. What we're doing is sharing the amount of time that an electron is around the nucleus of both atoms participating in the bond. So for example, on the screen, what I'm saying is 50% of the shared electron's time would be around the left carbon atom and 50% of the time would be around the right carbon atom. So we're sharing time. We're not actually cutting an electron in half. Now, I know most of you know that. Some of you have had chemistry, so that's an obvious statement. But there are some folks here that have not had chemistry, so I do need to say these sorts of things. Say what we're sharing is the time of the electrons. How much time is spent around the left atom? How much time is spent around the right atom? You also notice that with a nonpolar bond, notice that the nonpolar bond is between atoms that are identical, right? Notice that both atoms in the nonpolar bond are both carbon. Okay, that's significant. And what I'm suggesting to you is 9 times out of 10, you can predict whether a chemical bond is going to be polar or nonpolar just by looking at the identity of the atoms that are participating in the bond. If the atoms are the same, there's a very good chance the bond's going to have a nonpolar characteristic to it. If, on the other hand, as we go to the bottom, right, we wrote this up last time, we have a polar covalent bond. Notice that both atoms here are in fact different. Notice that both atoms are different. Okay, so if you have 2 different atoms that are participating in the bond, there's a very good predictability that the bond's going to be of a polar characteristic. Okay, now there's some things in chemical theory that can throw a wrench into things, but this is not a full-fledged biochem course. We're just talking about the basics right now. Okay, all right, so do you have any questions before I continue?
So not all bonds that are the same atom are going to be nonpolar, and not all bonds that are polar will have 2 different atoms, correct?
So Rachel, the quickest answer is yes, but it's like looking at a commercial, there's always some fine print. So what I'm saying to you is, as you get into the field of biology, as you've had more proper chemistry classes, what can throw a wrench into things is if we have, and I'm not going to draw it right now, but let's say up on the carbon-carbon nonpolar example that we're showing, what if there were extra atoms attached to the left carbon? What if there were extra atoms attached to the left carbon? What if there were extra atoms attached to the left carbon? Those extra atoms would have protons in them, correct? So they could also draw electrons from the right carbon towards the left carbon and start to make that bond have a more polar characteristic at that point. So just looking at 2 atoms, it's very predictable whether a bond's going to be polar or nonpolar. But if we look at 2 atoms that themselves have additional atoms attached, then the answer to whether the bond is polar or nonpolar gets a lot murkier a lot quicker. Okay.
Okay, thank you.
Okay, but that's a very good question to ask about that, right? There's theory and then there's reality, and sometimes there's way too much difference between those 2 things. Okay. So right now we're just giving you a way to begin to think about these topics. So Okay. Now, what I want to share with you is something else that's kind of subtle, but it's very important. Okay, let's take a deeper look at this polar covalent bond. We know from the way the information has been drawn on the screen that oxygen is pulling electrons towards itself more frequently, doing a better job of it. Oxygen is more of an electron vacuum cleaner than hydrogen. And so what that means is that most of the time, the oxygen atom, because it's better at drawing electrons towards itself, that means most of the negative charge characteristics of the electrons spending most of their time around oxygen will cause the oxygen atom to take on a partial negative charge. Okay, so I'm writing a weird little symbol here. This is a Greek letter, which refers to partial. A partial. So partial negative or partial positive. Okay. So the oxygen in a hydrogen-oxygen polar covalent bond is going to take on a slightly or partial negative charge because oxygen has more electrons spending more of their shared time around the oxygen atom. And hydrogen, it doesn't get the benefit from the presence of the electrons nearly as much, nearly as large a percentage of time. So the hydrogen takes on a slightly partial positive charge. Okay. All right, so let's see how we can do here. All right. Okay, so let's see who I can ask a question of here today. Okay, how about Chelsea Benzinger? Chelsea, could you unmute your microphone and say hi to us?
Hello.
Hi, Chelsea. Chelsea, got a question for you. In the polar covalent bond on the screen between hydrogen and oxygen, why is oxygen better at drawing electrons towards itself than hydrogen? Why is oxygen better at doing this?
Isn't it because it kind of has a higher electronegativity involved with it?
Well, you are correct. And that would be one satisfactory answer for my question. So that was a nice response, but it's getting into a different sort of vocab word that we haven't talked about. Would you be able to explain this differently? Or Chelsea, would you be able to explain why one atom might be more electronegative than another atom?
Let me see if I can kind of make this. Is it because it can attract several smaller, okay, it's like oxygen can attract more hydrogen to it? I really have no idea.
Okay. Thanks, Chelsea, for participating today. I really do appreciate it. And just, as a general example for everybody, the answer Chelsea gave about oxygen being more electronegative is correct. But, and I'm picking on Chelsea now, but that's what I do. It seems like Chelsea's understanding of what electronegative really means might be a little bit weak, right? So this is where we build up our education, our knowledge. That's why we're here. So Chelsea, and for everybody's benefit, what I'd like you to consider is that the reason oxygen is better at drawing electrons towards itself is due to the fact that the atomic number of oxygen is greater than the atomic number of hydrogen. So who can be the first to type into the chat window the atomic numbers of hydrogen and oxygen? What are the atomic numbers of hydrogen and oxygen? You should be able to find that in your textbook. Okay. All right. So Taylor was very quick on the draw there. So I love it. Fantastic. Okay. So thanks everybody for searching and looking these things up. Right. Oxygen has 8 protons. 8 positively charged particles, which are attracted to electrons, right? So oxygen has 8 protons that are tugging on their side of the tug of war rope, and hydrogen has 1 proton on its side of the tug of war rope. So if we have a tug of war and one team has 8 and the other team has 1, doesn't it make more sense that probably the team with 8 is going to win the tug of war for electrons? Chelsea, does this make sense?
Yeah.
Okay, great. Okay, so let's just make a comment here. Oxygen can more strongly attract electrons than hydrogen because oxygen has a larger atomic number. Okay. So my point in asking these sorts of questions, folks, is for you to realize that even with fairly basic rudimentary information as presented in the chapter of your textbook, you can really start to think about some fairly sophisticated aspects of chemistry. Okay. All right. Do you have any questions right now? Okay. So keep in the back of your brain this idea of polar covalent bonds and the idea that, for example, an oxygen atom would have a partial negative charge and a hydrogen atom would have a partial positive charge. This is going to come back around in our conversation today. Okay, so we're doing okay. Please fire away with the questions if you have them. Okay, so I'm going to move to the third type of bond, which is the hydrogen bond. Okay, a hydrogen bond is an attraction created by a slightly positively charged hydrogen atom and a slightly negatively charged atom, which is often oxygen, but not always oxygen. It is often oxygen, but not always oxygen nearby. Let me just read that to be sure I've typed it into your notes properly. A hydrogen bond is an attraction created by a slightly positively charged hydrogen atom, i.e. this thing up here I'm circling with my cursor, and a slightly negatively charged atom nearby. Okay, now there's a little bit of a nuance to this. So the way I like to describe this is in the context of starting a conversation about water. So let's do a little drawing with water, okay? All right, so here we go. H2O. H2O, there we go. So on your screen, we have, right here, we have one water molecule. All right. So these bonds here that I'm going to put in a blue rectangle. Okay, well, I want to ask some members of the audience here. Okay. Let's see. Subitra Biswa, could you say hi to us?
Hi,
Hi, Subitra, how are you?
good. Thank you.
Okay. Good to see you kind of online here. So Subitra, with the water molecule that I've drawn on the screen in front of you, can you tell me how many hydrogen bonds are present?
2 or 1?
Okay, so pretend we're on a game show right now and you have to give me a final answer. So is your answer going to be 2 or 1 or something else?
1.
Okay. All right. So
However,
Biswa, thank you for participating today. I really appreciate that. And let me ask a question in the chat window. Okay, do you agree with Biswa that there is one hydrogen bond displayed in this water molecule? Okay, so I'm getting some no's here. And I think I want to say thank you to everybody that's participating. So if you don't agree with Biswa, can you type a number that you think that represents the number of hydrogen bonds? Okay, beautiful, beautiful. Folks, thanks for participating. It really makes class so much better. I mean, isn't it a little better that you're actually doing something than just sitting here kind of listening to me for an hour? I mean, my God, that's boring, right? So let me say something, folks. The answer to my question is 0. There are 0 hydrogen bonds in this drawing. There are 0 hydrogen bonds in this drawing. Okay. Okay, so, all right. And so if that's puzzling you, let me see if I can unpuzzle your puzzler. Okay. So those bonds that I wrote in the rectangle, right? You know that these are polar covalent bonds. These are polar covalent because didn't we just talk about that up here? HO, 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 right? There's well, I don't want the drawing to get too ugly, but you see HO and HO, right? Both of those are polar covalent bonds. They are not hydrogen bonds. Okay. So now wait a second, Dr. Allen, what the heck are you talking about? Because it seems like this definition, I just drew on my own notes, seems like that definition should fit. Well, there's some nuance here. Okay. So let me see if I can make this better for you. Okay. Do you notice in figure 2.4, page 29 of your book, I hope you have that available. Do you notice that the hydrogen bonds are drawn in between water molecules? It's not the bond in between the atoms of a single water molecule. It's a bond found in between the atoms of at least 2 water molecules. So if I do this, let me draw another water molecule up here. Okay, and doggone it, I'm kind of out of space here. I messed myself up, but I'll do it this way. Hydrogen, oxygen, hydrogen. So here's a second water molecule. All right. And Kid, I'm looking at your comment on the chat window. And so the answer is yes. The answer is yes. Kid has a statement. So between the hydrogen atoms of one molecule and the oxygen atom of the other molecule, yes, Kid. Now there are exceptions. If you have a very large molecule, let's say an insulin protein, you could have a hydrogen atom on one end of that protein and an oxygen atom on the other end of that protein, that depending on how the protein bent and contorted and twisted its shape, you might get an intramolecular hydrogen bond. Those things do exist, that can happen. But it is much more common that the hydrogen bonds exist in between 2 different molecules. And that's what I'm trying to suggest right now, particularly as we start a conversation about water. So let's look at this again and figure out what the heck's going on. Remember that within one water molecule, the hydrogen and oxygen bond is polar covalent. So what does that mean? Well, here's what it means. It means that from our previous conversation just a little bit ago, it means that that oxygen is going to take on a slightly negative characteristic, and the hydrogen on the same molecule, slightly positive characteristic. That's going to happen on both water molecules, slightly negative down here, slightly positive here. And so what have we created? Haven't we created a scenario where we have an oxygen, we have the oxygen of the top water molecule with a partial negative charge sitting next to a hydrogen on the bottom water molecule with a partial positive charge. And what do we know from elementary school? Opposites attract, right? So in other words, if I have a partial negative and a partial positive, I will actually form a hydrogen bond there. Okay. All right. So I don't want to spend too much more time on this, but I'm happy to take any questions you might have right now. All right, so let me ask one more question in the chat window to try to get everybody's participation here. I'm going to give you a statement, and I want you to tell me if my statement is true or false. Let me read my comment here, make sure it's right. Okay, here we go. True or false? Hydrogen bonds in between water molecules cannot form if the atoms within each water molecule were to be joined by non-polar covalent bonds. Okay, I'm just waiting for more responses. Okay, please take a look at your chat window, look at the community of responses. And yeah, I agree with you folks. That is a true statement. So if you're really getting why the answer to that statement is true, then good for you, because that means you're really starting to appreciate what we're talking about here, right? Okay, if what is true, the polar covalent bonds within a water molecule happen to be nonpolar. They're not. In reality, they truly are polar. But if they happen to be nonpolar, there would be no way that the oxygen and hydrogen atoms would take on partial positive or negative charges. So we would not create the foundation for a hydrogen bond to form in the first place. It looks to me like you all that participated answered that correct. Nicely done. Okay. Do you have any questions? All right, so I might have to see if I can fix our notes later on here. Okay, let's see what we can do. At any rate, okay. All right. If there are no questions, folks, I'm going to put to rest the topic of chemical bonds. Okay, so thank God that's over, right? All right, so this is not a biochemistry course. I am not going to throw sophisticated chemical reactions at you on exams. I'm really not. You shouldn't worry about that. But the basic kind of knowledge that we're trying to discuss right now from chapter 2, well, this stuff is testable. This stuff is quizzable. I want you to think about these things. All right. So what I would like to do next is talk to you about, as we move through the chapter here, chapter 2, page 31 talks about inorganic compounds that are important for biological systems. And I would be remiss if I didn't talk about water. Okay, so I want to talk to you about the important biological properties of water. Okay, you've probably read this in other books and other courses. If you've had me, there's a good chance that I've talked about this before. But remember, even though we are reviewing principles here in microbiology, the point is that all the chemistry and the water stuff that you might have heard about in A&P is also legitimately correct for the field of microbiology. The rules of biochemistry do not change simply because we're talking about a bacterial cell versus a human cell. Things are pretty much the same. So let's list some of these important biological properties of water out. Okay, so we know that water is a fantastic solvent. Okay, and so for those that haven't been thinking about biology in a while, you remember that a solvent is something that's very good at dissolving material. Okay, and so, it is extremely important, for all cells, including bacteria or fungi or protozoa or algae or whatever you want to consider. It is very important for all cells that various macromolecules that are very large, such as starch and other things, can be broken down into smaller components so that the enzymes of the cell can nibble on these things and produce energy so that the cell can live. I mean, that's basically what's happening here. So please be aware of this equation, this statement: a solution equals a solute plus a solvent, right? And so just quickly here, as an example, if we have a cup of unsweetened coffee, well, then I could ask a couple questions here. If we had a cup of unsweetened coffee, okay, so we didn't add any sugar to it or anything like that, okay, the question could be, what is the solution here? Well, the solution in this example is the cup of coffee. It is the coffee, okay? Question 2, what is the solvent? Well, the water of the coffee, right? I mean, coffee is a liquid, right? We think of it as probably a brown or black liquid, but it's basically colored water, is it not? It is colored water. So the water of the coffee is the solvent. And what is the solute? Well, the solute is going to be the thing that turns the water from clear to some kind of brown black, right? So I'm talking about the pigments from the coffee beans that were used to actually brew the coffee. Okay, pigment molecules from coffee beans used to brew the coffee. All right, so we don't have to go crazy with this, but I just want to be sure you appreciate some of this common language. Okay. Okay. Please stop me if you have any questions. I'll be happy to chat about anything that's bothering you. Okay. All right. So water is a fantastic solvent. Number 2, okay. Water is a fantastic temperature buffer. Okay. And by temperature buffer, what I mean is that water is very good at resisting changes in temperature, right? Be very careful, particularly new students. You see that word buffer and you're automatically thinking about pH, because a lot of books talk about, buffering systems when you're talking about the concept of pH, but this is not pH, this is temperature. So I'm talking about a temperature buffer, okay? A buffer is anything that resists change. In this context, we are saying that water is very good at resisting changes in temperature. Okay, all right. Okay, so let's see here. I'm just going down my list here. Victoria Freeman. Victoria, could you say hi to us, please?
Hello.
Hi, Victoria. Hey, Victoria, I don't know if what your science background is, but, why would it be helpful that the water inside a bacterial cell helps maintain a constant temperature environment? Why would that be helpful?
I know bacteria cells are like bad, but maybe to keep it from not dying or something like that. I'm not sure.
From not dying. Okay, well, that is a true statement, Victoria. You haven't said anything wrong there. I mean, that's good. Now, let me pick on a little bit of your other language. You said to me, I think you said to me that bacteria are bad.
Well, I know they can be good sometimes though, right?
Yes, absolutely.
Yes.
And that's what I'm going to bring out, right? We tend to vilify those things that harm us, right? So,
Mhm.
Streptococcus pyogenes, the bacterium that causes strep throat. Oh, very bad, because it makes us sick, right? But on the same, and on the other side of the coin, we have bacteria like E. coli and so forth inside our intestine that ensuring we can absorb vitamins from the foods that we eat in our diet. So, it's our natural tendency to vilify things, but we got to appreciate there's a lot of good that microorganisms do for us. Victoria, thanks for your participation today.
You're welcome.
So I'm going to put this question up here again. Why is it helpful to a bacterial cell that the water inside it helps maintain a constant temperature? Okay, now Victoria did say so that the bacterium doesn't die, and that's true. Okay, so we could start there. And so I want to go further with this. Why would the bacterial cell die? Why would the bacterial cell potentially die if the temperature changed? Okay, so Chelsea has an interesting statement in the chat window. So proteins of the cell don't denature. Chelsea, could you unmute your microphone for me?
Sure.
Chelsea, that's a very nice statement. Could you explain to everybody what denature means?
Sure. So when a protein heats up too much, kind of like in a fever, the protein structure will actually unravel, causing the protein no longer to work.
Okay, you said a lot of good things and some fairly sophisticated language. I love that. So thank you. I will say that I tend to want to talk in perhaps some simpler terms for brand new students. So Chelsea, I'm going to take your idea and just change the language a little bit, but you did great. Okay. So basically what Chelsea is saying is that if you change the temperature that molecules are in a cell, the molecules will change their three-dimensional shape. They will change their shape. And if a molecule changes its shape, it will change how it behaves. It will change its function. So if a cell experiences a temperature change that it cannot regulate, the molecules in the cell will change their shape and change what they do to the point that the molecules may no longer actually function correctly and a cell could die from that. Okay, so everybody's doing a great job here. Kit's statement on the chat window is absolutely true. Okay, why would the bacterial cell potentially die if the temperature changed? Because changes in temperature will change the shape of molecules in that environment, including inside a bacterial cell. When molecules change shape, they change function, they often don't work properly anymore. Okay? This
Can
is
they
normal.
change back?
Can they change back? Who asked that?
Rachel.
Rachel. Okay, Rachel. They can change back to a degree. It's kind of like this, Rachel. It's kind of like having a rubber band. You could stretch a rubber band so far, and as long as you don't overstretch the rubber band so that the rubber of the rubber band starts to tear, then yes, the rubber band can go back to its normal healthy size and dimensions. So can molecules go back to their healthy shape? Yes, up to a point. It depends how badly they have had their shape deranged by the change in temperature. Other questions? Okay, so we know that water is a very good solvent. Water is a very good temperature buffer. Plenty of other things we could say, but I'm going to say one more thing and use that as a springboard into a different related concept here. And that is that water has the ability to influence pH. Water has the ability to influence pH. And while I wish I had the luxury of a little bit more time with you folks, but I got to pick and choose my battles here. Okay, so we're going to talk about pH next, but I want to just address this one thing. Water has the ability to influence pH. Well, the question is, how? How does water influence pH? Okay, and here's the answer. Water influences pH by being the solvent that dissolves acids and bases. Okay, water influences the pH by being the solvent that dissolves acids and bases. Okay, so if this is still a little murky to you, I'm going to try to help you understand what pH is all about next. Okay, so we're going to talk about pH next. Okay, all right, so our last major talking point was the important properties of water, and we ended with this idea about pH. So I'm going to make another bullet point here, the pH and the pH scale. Okay, so we're moving forward here in our notes. And the pH scale, excuse me, as you see, is on page 33 of your book. Page 33 of your book. I bet you know all about this. I bet you've heard of this before. Let's just define a couple things. pH is a scale used to measure the hydrogen ion concentration of a liquid, okay, which, honestly, typically that's going to be water, okay, and when we talk about liquids in biology, 99.9% of the time we're going to be talking about water. We're certainly not going to be talking about antifreeze. That would be lethal, okay. So pH is a scale used to measure the hydrogen ion concentration of a liquid like water. You can see in your own textbook, figure 2.7, that, the pH scale is represented from 0 to 14. And just for conservation purposes, I'll put 0 here, and then we'll go the whole way out to, we'll go to 7, which is basically neutral, and then we'll go to 14 here. And we know that down by the 0 range is where the solution would be acidic. So we have acid on the lower range. Okay, we know that it's basically neutral in the middle. And we know it's basic with the higher pH values around 14. Okay, all right. So this gets us started here, and you can see some of the pH values of common liquids we might encounter in daily life, right? I mean, take a look there. You have pure water, which is 7. The physiologically healthy pH of human blood, which you might remember from your A&P classes, is about 7.35 to 7.45. If I go below 7, we have urine there at a pH of 6, wine at a pH of probably around 4, and so on and so forth. Take a look at stomach acid, right? Stomach acid in your belly right now is powerfully corrosive acid, typically a pH of around 1.5. So at any rate, you don't have to memorize those values, but I do want you to think about what pH is and why this is important, okay? So perhaps I should back up. Perhaps I changed from my water conversation too quick. So water has the ability to influence pH. How? By acting as a solvent that dissolves acids and bases. Why is the topic of pH important to all cells, including those in microbiology? Folks, I'm just going to give this to you, okay? I'm going to give this to you. Just like we stated a little bit ago, if you change the temperature of the environment that molecules in, they change their shape. Well, in an analogous statement, if you change the pH of the environment that molecules are in, they will also change their shape, okay? Now, this says relevance. This does have relevance here to disease processes with microorganisms, and I'll try to come back to that in just a little bit. Yeah, Kit says all these free hydrogen ions could cause some chemical reactions in the cells. It's absolutely true, Kit. It could trigger chemical reactions, or it could suppress chemical reactions. So absolutely true. Okay. All right. Okay. So let's see. We should define acids and bases, shouldn't we? Okay, because I talked about acids and bases up here, we should at least define them for you. Okay, so an acid is any molecule that when dissolved in water releases some hydrogen ions. Okay, any molecule that when dissolved in water releases hydrogen ions. Take a look at that definition. Do you notice that the word water is part of the definition of acid? My point is this, that an acid cannot act like an acid. It cannot behave like an acid unless it is in the presence of water. Okay, that's in a very silly way. That's kind of like saying you cannot behave as a daughter unless you're in the presence of your mom and dad. But this is true here. An acid cannot behave as an acid if water's not around. Okay, so for example, okay, HCl, this is hydrochloric acid. Well, that's nice, but hydrochloric acid can't do what an acid does unless water's present. Okay. Number 4. A base. Okay. A base is any molecule that when you dissolve it in water, it will bind hydrogen ions. It will attach to them. It will soak them up like a hydrogen ion sponge. And by doing that, you lower the concentration of hydrogen ions in the environment. Okay, let me make a statement here with the acid. You will increase the hydrogen A base does the opposite. Any molecule that when dissolved in water will bind to free hydrogen ions, thereby decreasing hydrogen ion concentration in the solution. Okay. All right, so this sounds all very good and technical and theoretical, Dr. Allen, but why the hell do I have to know this for microbiology? Let's give you a clinical application, okay? So 5, clinical relevance of pH to disease in humans. Honestly, it's true for animals, all animals as well. But okay. All right, let me ask you a question in the chat window. Is there anyone in your life that is diabetic? Okay, I picked diabetes because it's a pretty common disease state, right? I hope you're not diabetic, but you know, if you are, you are. I mean, that's just how it is, right? So, but we kind of know folks that are diabetic, and we know that they probably take insulin to regulate their blood sugar and all that kind of good stuff. You know, we're not going to go into physiology right now, but it's a very common disease. It's a very debilitating disease. And before mankind developed sufficient technology to purify animal insulin decades ago, diabetes was a devastating disease because people would often develop, gangrene in their limbs, and they would have to have their limbs amputated, things like this, right? In fact, one of my great aunts had to have her legs amputated due to diabetes, so not pleasant at all. But at any rate, the clinical relevance of pH to disease in humans, let's consider diabetes. Okay. Now, I don't have time to go into a big discussion of this, but here's what I want to present to you. Folks with diabetes that are not controlling the disease often have a bloodstream that's too acidic. Okay, so why is a diabetic's bloodstream, a diabetic that's not controlled with medication, why is their diabetes create excess acid in the bloodstream? And it's because when a diabetic patient's cells get confused about blood sugar regulation, the cells start to burn triglyceride molecules that are stored particularly in fat tissue. And when they burn the triglyceride molecule, they throw out fatty acids into the bloodstream, which acidifies the bloodstream. Okay, so fatty acids from fat cells are inappropriately deposited into the person's blood. Okay, so long story short, if you're an untreated diabetic, you're going to have an acidic bloodstream because of this problem with fatty acids. And so what is the clinical relevance here? Okay, if you change the pH of your body fluid, then microbes that would not normally grow under healthy blood pH will start to grow. There it is, folks, right? Our healthy pH in human blood and our body fluids is roughly 7.4, okay? And generally speaking, that is sufficient to protect us against the undesirable growth of certain microorganisms that might cause us harm. But if you acidify your body fluids, all of a sudden microbes that don't normally cause you harm now will cause you harm. Okay, so an example of this would be diabetics struggle with overgrowth of fungi, particularly yeast, that can damage their organs. Okay, so the take-home message I'm trying to get to you is that even though chapter 2 is very theoretical and you think, oh, this is just crap I got to learn for school because I got to learn it for school, it does have clinical relevance. If you have any disease state which alters the pH of a body fluid or a surface like your skin, then what that will do is start encouraging the growth of microorganisms that are normally suppressed by the healthy pH of body fluids and skin. Okay, so let me just show you something. Let me see here. Hold on a second. Let me just show you something here. I don't know how much experience you guys have yet on doing some forms of research in biology, but you're going to get there someday. At any rate, there's this place called the National Center for Biotechnology Information. It is a computer database that's funded by your US taxpayer dollars, and it's got a lot of important biomedical research information. Long story short, I just want to do something. I want to type into this database, into their PubMed database. I want to type in diabetes and fungi and see how many articles are returned by doing this. So here we go. 1, 2, 3, bang. Oh boy, there we go. Okay, take a look at this. 24,548 publications with the two terms of diabetes and fungi. Okay, urinary tract infections in patients with diabetes mellitus, diabetic foot ulcers. You know, my mom was a diabetic. She struggled with foot problems, you know, while she was alive, that kind of thing. And the point of it is, folks, that a lot of research is involved with microbial infections that start because the pH of the body fluids is altered by a disease process. Okay, so pH is extremely relevant because it will support the growth of microorganisms that are damaging to our bodies. Questions? Okay, I'm determined to get this chapter finished here. So we're going to move on to the topic of the organic macromolecules. It's introduced as a heading at the bottom right of page 33, but then it spills over into basically the end of the chapter. Okay, gang, I will never ask you to draw out a chemical. I will never ask you to draw out a chemical equation or the chemical structure of a molecule. I'm not going to do that. Okay, but you should have some basic familiarity with the different classes of molecules inside cells that are not water. I mean, that's basically what we're talking about here. So we're going to talk about the 4 major categories of organic macromolecules. All right. And so first off, when you say organic macromolecules, you might say, well, what are you talking about? Well, I'm talking about large molecules that are not water. Large molecules that are not water, okay? Right? We know that 65 to 70% of the mass of a cell is going to be due to the presence of water. So water is extremely significant. Okay. But if we ignore the water for a moment, what else do we find? Well, we find 4 major other types of molecules that are not water. Okay. The term organic refers to the presence of molecules that are built off the linking of hundreds to thousands to tens of thousands of carbon atoms. Okay, so I'm using the word organic differently than you would use it at the grocery store, right? People think at the grocery store that organic means natural and everything's nice and healthy and blah, blah, blah. Yeah, okay, that's okay for everyday life. But organic to a scientist, specifically to a chemist, tells you that carbon atoms are present. That's the big deal. Carbon atoms are present. Okay. In fact, the chemistry of carbon atoms is so important that most undergraduate education has, if you're going to get like a bachelor's degree in biology, you would take a full 2-semester, 1-year sequence of organic chemistry, because it's that important if you're going to understand the chemistry of life. Well, we can't do that in 15 minutes here, but I can introduce you to some main ideas. Okay, so the major types are this: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, lipids, proteins, and nucleic acids. Most of the major structures we might talk about in a typical cell, whether it be a eukaryotic cell or a prokaryotic cell, most of the major structures are going to be assembled by linking varieties of carbohydrates, lipids, proteins, and nucleic acids together, or taking a look at how the machinery of a cell decodes information that's present in these macromolecules. Okay, so again, I really don't care that you can draw out any chemical structures. That's not important for this course, but you at least have to be familiar that these things exist. So let's talk about each of these just a little bit. So first we have carbohydrates. Okay, carbohydrates, there's many classes of there's many different types of carbohydrates. And you might fit them into this sort of sequence, right? We have monosaccharides, disaccharides, trisaccharides, right? And we can keep linking them together. We can put 4 together, 5 together, 10 together, 100 together, 1,000 together. So eventually we get up to what we think of as a polysaccharide. Okay, a polysaccharide. So let me ask a couple questions here in the chat window. What is the primary storage polysaccharide in plants? Okay, thanks for guessing, Kit. It's not a chloroplast. Chloroplast is an organelle, but that was a nice idea. Chelsea, I want to run with yours. It's starch, right? Starch is the major storage polysaccharide. Right, and I bring that up because probably many of you had some French fries over Labor Day weekend, and of course potatoes are plants. All right, what's the storage polysaccharide in animal tissue? Well, I'll give this one to you, right? It's glycogen. Okay, the point of it is that carbohydrates are used for, many different purposes. In microbiology, we're going to encounter polysaccharides in the cell walls of bacteria and fungi. Okay, we're going to encounter polysaccharides in the cell walls of bacteria and fungi. It's a future talking point for me in chapter 4, which we're headed to very quickly. Okay, so gang, be sure you are aware of carbohydrates. Be sure you know what a monosaccharide is. Okay, but don't be worried that I'm going to ask you to draw out the structure of one of these things. I'm not going to do that. Okay. All right. Do you have any questions? All right, the next major category of organic macromolecule are your lipids. Okay. Lipids, you see some chemical structures of lipids on page 38 and 39. So there are multiple types of lipids. Okay. And certainly the list that I'm giving you is not exhaustive, right? But, when we talk about lipids, we talk about fatty acids, Okay, so I just want to be sure you're with me, gang. I just want to be sure you're with me. So let's do a little quiz here from your textbook. Here we go. I typed in it too fast. On figure 2.9, part B, what fatty acid is drawn for part B? What fatty acid is drawn on page 38, figure 2.9, part B? Okay. All right. Thanks, folks. Appreciate it. So Chelsea, Erica, you guys and gals got it right off the bat. Good job. Palmitic acid. So here's the deal. Am I going to ask you on an exam to draw the structure of palmitic acid? No way. I'm not going to do that. I mean, if this were a biochem course, I might, but not for a beginner micro. But the point of it is, do you know what a fatty acid is? If you saw a picture or diagram of it on an exam, could you say, oh yeah, that's a fatty acid? Could you do that? That could happen. Okay. So we have fatty acids, we have triglycerides, we have phospholipids on page 39, which many of you know create the plasma membrane of our cells, including bacterial cells, fungal cells, you name it, phospholipids are it. And then we also have steroids, of which cholesterol is a typical example. You see at the bottom of page 39, okay? So you're going to very commonly see lipids as part of the plasma membrane makeup of microbes. Okay, we're going to be talking about this in chapter 4. Okay, don't try to memorize these chemical structures, but certainly you got to know what I'm talking about. If I talk about a phospholipid, okay, you have to know what I'm referring to. Okay, racing right along here. Next we have proteins. Okay, so proteins, well, what can we say about proteins, right? I mean, entire textbooks have been written on carbohydrates. Entire textbooks have been written on lipids. Entire textbooks on proteins. Entire textbooks on nucleic acid. So all we're doing is dipping your toes into the water here. But I'm saying you do need to pay attention to these topics when you study. proteins are used by cells for many purposes. For example, some proteins are enzymes, right? So enzymes accelerate chemical reactions. That's going to be a big topic for us when we talk about chapter 5, which is metabolism. Some proteins are receptors, right? Some proteins are receptors. So that means they bind things in the environment and maybe internalize things into the cell, right? How in the world would a Vibrio cholera cell bring a glucose molecule inside the cytoplasm? Well, maybe there's a glucose receptor. Maybe there's a protein that brings that monosaccharide into the cytoplasm. Some proteins are structural in nature. Okay, they will be used to build things, such as the cell wall of bacteria. Right, cell wall of bacteria includes protein. So proteins are used by a lot of things, and proteins are always made of amino acids. Right, proteins are always made of amino acids. You have table 2.5 on page 41. Okay, do not try to memorize those chemical structures. I don't want you to. If this were biochem, let me put it this way. I did not personally have to memorize the amino acid structures till I got to graduate school. So we don't typically ask it of beginning students yet. So just be familiar with what an amino acid is, but don't try to memorize their structures. I'm going to ask a question here, and I can see I'm probably going to need a few minutes in lab for some continuing discussion here, but is Ireland online with us? Ireland?
Hi.
Hi, Ireland. Ireland, do you have your textbook open?
Yes.
Okay. Could I ask you a question, please? And this is for everybody, but I'll ask Arlen, see how she does with it. Arlen, on page 41, if you look at the very first column of that table, you see 4 amino acids. They are glycine, serine, aspartic acid, and phenylalanine. So my question to you, Arlen, is how can you expect aspartic acid to behave inside a cell?
Yeah, I don't know actually.
Okay, so Arlen, let me try to lead you to the answer here. Okay, so Arlen, what is the most common substance we find inside a cell?
I mean, we're talking about amino acids, so I'm assuming it has to do with that.
Well, for right now, step back and just take a deep breath. Don't, just imagine that we're having a casual conversation. Nobody's looking at you, and your grade does not depend on this. What have we said is the most common substance that we talked a lot about today that's found in water?
Water?
Damn, I gave it away. Okay.
I was getting there, and then you said the W, and I was like, there you go.
All right, so it's water. Okay, so Ireland, in the context of this, how does aspartic acid behave inside a cell? I'm asking you, what should we expect to happen to aspartic acid if it's inside the watery environment of a cell?
Would it be kind of like the same thing we were talking about earlier with the acid and like the pH and stuff with water?
Yes, so tell me
Okay,
what's going to
so
happen.
it would, when it's with water, it would dissociate, and there would be like hydrogen atoms.
Okay.
Is that okay?
All right, so let me ask it very bluntly, Arlen. Is aspartic acid going to increase the hydrogen ion concentration inside the cell or decrease it?
Increase?
I think you said increase, and you win the prize for today. Good job, Arlen. Okay. All right. Yes. So how does aspartic acid behave inside a cell? Well, the answer is it will behave like any other kind of acid. It will release some hydrogen ions, which increases the hydrogen ion concentration. My point in asking this question is not to seem obtuse, but to get you to see that just because we start thinking about proteins, or just because we start thinking about cells, doesn't mean you should throw away all your knowledge of chemistry that you're working so hard to study and understand for the course, right? Aspartic acid might be a building block of a protein, but aspartic acid is still a molecule. It happens to be an acid because of its behavior in water. So what do we expect it to do when it's in the cytoplasm of a cell? It's going to release hydrogen ions, okay? So my point is, don't throw out all that glorious information you have in your head simply because we're slightly changing the topic. That has to stay in your head so you can reason with it. Okay, folks, I better stop here. And we have lab at 2 o'clock, so I'll leave the Zoom channel open, okay? So just go take a quick bathroom break, rest, and I'll see you in 15 minutes, okay? Hello, everyone, and welcome back.
Hello.
So, I think what I'll do is take maybe 30 minutes of today's lab to get us in good shape with our lecture, okay? And then we can go over any concerns you have about last week's lab, and we can introduce this week's lab sort of homework to you, okay? So we should be able to accomplish all of that by 3:30, I think. All right, do you have any questions at this time? Okay, thank you, Elena. Okay, well, let's finish up talking about chapter 2 and at least get chapter 4 started. Okay, so we were trying to at least put a quick list together of the various categories of organic macromolecules that we find in all cells, including the cells of microbiology that we talk about and that we care about: carbohydrates, lipids, proteins. The last category is nucleic acid. Nucleic acids. And so let me say this. I bet you have all heard of the various types of nucleic acids, okay? So nucleic acids have two major types, and that would be DNA and RNA. Okay, so here we go. Question in the chat window. Here we go. Without looking it up, can you spell the full word that is represented by DNA?
Deoxyribonucleic acid.
You bet, you bet. Okay, so Taylor has it there, right? I mean, there's a little bit of a spelling issue, but it's basically right. Okay, so my point in asking that is we've gotten so used to thinking about DNA and RNA that we don't even remember what that means anymore, right? So DNA is short for deoxyribonucleic acid, right? So deoxyribonucleic is one word, acid is the second word. RNA is ribonucleic acid, okay? So whether I'm talking about DNA or RNA, all DNA and RNA are made of these fundamental little building blocks, kind of like amino acids are the building blocks of protein. Nucleic acids, DNA and RNA have a special kind of building block called a nucleotide. All DNA and RNA is made of the basic building block called a nucleotide. Okay, called a nucleotide. And so you have examples of a nucleotide, figure 217, figure 217 on page 46. Figure 217 on page 46 is an example of a nucleotide commonly found in RNA called uracil. Okay. And so right now, as I say, we're just dipping your toes into the water and pointing out things you want to pay attention to, be aware of for your first lecture exam. And then, you'll be good to go. Let me see here. We'll take a quick look at your syllabus. Okay, all right. So next week, I'm going to have to make a decision about whether we keep exam 1 on the 21st or whether we move it a little bit. I'm unsure yet of how that will pan out. I am pushing very hard to get us through the first 3 chapters of the course, because as I said, chapter 1 is a history chapter, chapter 2 is chemistry chapter, which if you've had any other bio courses should be a kind of a review. So the only really new chapter is chapter 4. So I want to get us into the semester and get a basic exam taken care of to be sure that you're feeling confident about things. And then we can actually begin the real course. The real course actually begins, in my opinion, in chapter 8 when we talk about genetics. And genetics will heavily involve a discussion of nucleic acids that we have on your screen right now, DNA and RNA. So we will go into a lot of detail when we get to chapter 8. Okay. Gang, do you have any questions that you would like to ask about anything in chapter 2? Otherwise, I'm going to tuck this away. Okay, I'm not hearing anything, so hopefully that means you are feeling comfortable, or through some private study, you will feel comfortable. Remember, you can always pop in during office hours and say, hey, what about this, Dr. Allen? Don't forget, we do have tutoring available for this course. Remember, I can show you how to get to the tutoring if you want that. That's free to you. You don't have to pay any extra money for it. Okay. Let's close the page on chapter 2 and let's start the final chapter of content that will be on the very first exam, which is chapter 4. Starts on page 72 of the current edition of the textbook. Okay. Chapter 4 it is. Now, in some ways, some of the content of chapter 4 will be reviewed for many of you that have had any other biologies, because we will at least mention some eukaryotic cell anatomy. So let's be honest. If you've had any other biology course, I bet you've heard of the word nucleus, right? The nucleus of a cell. So from that perspective, this is kind of review. What will not be review for many of you is a special emphasis on the anatomy of prokaryotic cells, like bacteria. Okay, so there will be, I expect, some new things here that maybe you have heard of, maybe some things you haven't heard of. Okay, All right, so the topic of chapter 4 is functional anatomy of prokaryotic and eukaryotic cells. You already know, at least I think you do, right? There's two primary types of cells on the planet. We've listed this in your notes. They are prokaryotic cells and eukaryotic cells. And microbiology books like the one that you have and own and so forth used to make a really big deal about all these differences between prokaryotic cells and eukaryotic cells. And then as research continued and went on and on and on, it started to become clear that maybe there weren't quite as many differences as we thought there were. So the division line between prokaryotic cells and eukaryotic cells seems to have gotten smaller and smaller all of the time. That being stated, the one thing that you can very cleanly still say that distinguishes prokaryotic and eukaryotic cells is the presence of a nucleus. Okay, so prokaryotic cells do not have a nucleus. Okay, now remember from our conversation in chapter 1 that prokaryote includes bacteria and archaea. Bacteria and archaea. Just because bacteria and archaea don't have a nucleus does not mean they don't have any genetic cargo, right? A nucleus is simply a membrane-bound compartment in eukaryotic cells that wraps around the major chromosomes of the cell. Prokaryotic cells have chromosomes, they just don't have a membrane wrapped around it. Okay, so prokaryotic cells do not have a nucleus, but they still have DNA and RNA. Okay, eukaryotic cells do have a nucleus. Okay, they have a membrane wrapped around their genetic cargo. All right. And so what I'd like to do is put a table into your notes where we compare some of the commonalities and highlight some differences between prokaryotic and eukaryotic cells. So let's generate a comparison and contrast table. All right, and so I just need to take a look here. I need 2 columns and maybe about 8 rows. Okay, I can always change that if I need to. So we have prokaryotic cells. So remember, we're talking about cells in this chapter. I am not talking about viruses, right? Because viruses are not cells. We will have our conversation about viruses when we get to chapter 13 in the course. Okay. Actually, I already need to modify my table. I need to insert a column. Okay. So we'll get there yet. Okay. So in the leftmost column, I'm going to put some kind of structure or characteristic. Okay. So the very first thing I want to talk about is the nucleus. You already know the answer to that. Okay, so number 1, Is the nucleus present? Okay, we'll put nucleus with a question mark. Prokaryotic cells, the answer is no. There is no nucleus. Eukaryotic cells is definitely yes. Okay. Number 2, organelles. Do prokaryotic cells have organelles? Do eukaryotic cells have organelles? Okay. Remember, an organelle are membrane-bound inner compartments of a cell, with some arguable exceptions. Generally speaking, the answer for prokaryotic cell is no. No, with maybe some exceptions. And for eukaryotic cells, the answer is yes. Without a doubt, eukaryotic cells have things like mitochondria. They have lysosomes, right? They have the Golgi complex. If it's a plant cell, we're talking about chloroplasts, right? So again, eukaryotic cells definitely have organelles. Okay, the size. What is the typical size of a prokaryotic cell? Well, I can't give you one number and say that's always the size, right? Because there's too much variety of life on the planet. But let me give you a range of size, of which maybe you're starting to pick up on some things from your lab activity this past week. That's the whole purpose of doing this kind of stuff, is that the general range for a typical prokaryotic cell will be 0.2 to 2 micrometers. 0.2 to 2 millionths of a meter, right? A micrometer is 1/1,000,000th of a meter. You may not have it memorized yet, but by the end of the semester, you will have it memorized. Okay. Typical size for a eukaryotic cell would be anywhere from 10 to 100 micrometers, depending on the kind of cell that we're actually talking about. And again, you know, this is a general range. We're talking in generalities right now to have a nice comparison. There's always some exception that messes with your description, right? So just be a little bit open-minded about what we're saying here. Okay. Number 4, the fourth characteristic. Does the cell have a cell wall? The answer for prokaryotic cells is that usually a cell wall is present. Yes, usually, with some exceptions. Usually with some exceptions, okay? The cell wall in bacteria is made of a substance known as peptidoglycan, which will be a talking point for us later in this chapter. In eukaryotic cells, the answer that the best generalization I can give you is, do eukaryotic cells have a cell wall? The answer is sometimes. Sometimes. Okay, it's very contextual. It depends on the kind of cell we're talking about. For example, fungi definitely have cell walls. Plant cells have cell walls. But you know what? Our good old human cells of our body, we do not have cell walls. Okay, our cells do not have cell walls. Okay. And so this is an extremely important idea that I'm going to put down right below the table, and I'm going to emphasize it throughout the entire course. A cell wall is not the same thing as a cell membrane. A cell wall is not the same thing as a cell membrane. They are biochemically distinct structures. And I can say this 50 times, and you might hear me say it 50 times. And guess what? Somebody's going to tell me on an exam that a cell wall is a cell membrane. They are not the same thing. Okay, so number 5. Let's talk about, a cell membrane, which is also known as a plasma membrane. Okay, the answer for all kinds of cells is a big yes. Okay, both prokaryotic and eukaryotic cells have plasma membranes. Not all cells have a cell wall, because cell walls are not the same thing as a cell membrane. Okay. So the plasma membrane in prokaryotic cells typically has a very low steroid content. And the plasma membrane in eukaryotic cells typically has a high steroid content. As you know, steroids like cholesterol and things tend to be stuck into the phospholipid bilayer of the cells to enhance the flexibility of the plasma membrane. Okay, some other things we could say here. Number 6, do prokaryotic and eukaryotic cells have ribosomes? That's a big yes on both accounts. A big yes on both accounts. And let me say, this is something we'll talk about later in chapter 4. And when we talk about ribosomes, which you might remember from other courses, is the machine that makes protein. Ribosomes, they're described by biophysical experiments, which I will describe to you at a later date. But when you describe a prokaryotic ribosome, we simply describe it as a 70S particle. A prokaryotic ribosome is described as a 70S particle. A eukaryotic ribosome is described as a 80S particle. Okay, you might be familiar with that. If you're not, it's okay. Your book will introduce you to it, and I will talk about it later. Okay, let's continue here. Number 7. Number 7, mitochondria. Does the cell have mitochondria? Well, you know the mitochondria is the energy production power plant of a cell, but it might be surprising to you that prokaryotic cells do not have it. Prokaryotic cells do not have mitochondria. Eukaryotic cells do have mitochondria. I have a question I want to put in the chat window for you. Here we go. Do you know why bacteria do not have mitochondria? Do we have any sleuths in here? Anybody that's encountered this before in your background? Okay, Tiffany says because that is how they are made. Okay. Yes, all right, all right. Anybody else? Doesn't the mitochondria filter something? I'm not sure what you mean by that. In a way, it does filter hydrogen ions. Anybody else? Okay, so this is some fun stuff that we'll get to talk about at a later date, but let me give you the punchline here. Bacteria do not have mitochondria because bacteria are the mitochondria. They don't have mitochondria because they are mitochondria. All right. So we'll talk about that at a later time. Taylor, you mentioned that they produce energy another way. you might be surprised about that, Taylor. What I'm trying to get you to see is that in eukaryotic cells, the evidence suggests that mitochondria are symbiotic bacteria that live inside our cells. The scientific evidence suggests that mitochondria in eukaryotic cells, like our own cells, really are symbiotic bacteria that live inside our cells. So the mitochondria in your human cell is actually a bacterium. That's what the evidence suggests, okay? We'll talk more about that at a later time. It's kind of an intriguing story, and your textbook does talk about it towards the end of the chapter. Okay, let's see here. What else to say? All right, let me put another row in our table. And I think I'll probably stop with this, which is to say, okay, what about cell reproduction? Well, obviously both categories of cells, prokaryotic, eukaryotic, they need to be able to reproduce. Let me pause. There was a question from Kit. Is there evidence that a mitochondria could undergo binary fission and multiply? Kit, emphatically, yes. There is strong evidence for that. Okay. All right. So cell reproduction, yes. But the actual process of how the reproduction takes place is quite a bit different between a prokaryotic cell and a eukaryotic cell. Prokaryotic cell will perform something known as binary fission, which will be a talking point of chapter 6. And eukaryotic cells, well, obviously it's yes, but they use what you know to be true, mitosis and meiosis, right? If you recall your basic bio, mitosis and meiosis. You know what? I can't resist. I got to do one more row here. I got to do one more row. And that is on the topic of the cytoskeleton. Okay. You might have heard about the cytoskeleton. Cytoskeleton is basically the inner structure of a typical cell. It's kind of like the road network of tubes inside a cell that you can use to move cargo along. For a very long time, I mean, in terms of how long science has been happening, the scientific community believed that bacteria, prokaryotic cells, did not have a cytoskeleton. And we learned, I'm going to estimate 20 years ago, that that is in fact incorrect. Bacteria do have a cytoskeleton. Yes, they do have a cytoskeleton. Eukaryotic cells also have a cytoskeleton. They are biochemically distinct and different from each other in how they're made up, the molecules that make them up, and how they're arranged. But yes, both prokaryotic and eukaryotic cells do have a cytoskeleton. Okay. All right, folks. That's basically a half an hour, but it makes me feel better that I actually started chapter 4 today. So I'm going to stop there. Are there any questions you'd like to ask? Okay, so you take a look. Okay, all right. Okay. Okay, all right, folks. So I'm going to stop right there with my lecture for today. I'm also going to stop the recording. Okay.